So I'd like to get us started with some questions, and we do have some interesting questions for both Carl and Christian. And Thomas, I think you should feel free to come in on any of these points as we go through this discussion. A question perhaps first for Christian on benchmarking. How do you go about validating or understanding the predictive power of your platform to suggest what will then become active molecules once they're synthesized and testing? How have you benchmarked your platform to enable that?
Yeah, that's a great question, and in general can be called the general quest for predicting activities, and it totally depends on the scenario that you're working in. If you are in the lucky situation where you have a target structure, actually, you're much better off. You can do pretty precise and detailed compound minimization and affinity calculations. We at Biosolve IT have one such affinity scoring. It's called height scoring. Obviously, other methods from other companies are available there too. I won't go into the details of the pros and cons of the different approaches. I think that's a separate discussion. How to benchmark it then is obviously very difficult too, because what it would afford is try out all the available methods and then have all those compounds suggested, synthesized, and tested. Typically, not even the biggest pharma companies are at liberty to do such a broad spectrum of experiments. So what we all sort of fall back to is retrospective analysis. So certain molecules are active, and you try to re-predict their affinity, and in a virtual screening scenario, redetermine that they are really the best you can find in a particular data set. And increasing the size of the haystack, to come back to one of the phrases I used a lot in my presentation, doesn't change much here. The virtual screening just gives you the top candidates from the list, and the predictive power of the affinity calculation at the end remains the same challenge as before. So I wouldn't say that the larger spaces create a particular challenge here. I like to think that it just opens up many more opportunities. And it is not so important that every one of your prediction is correct. It is more important that you find some interesting needles. As I showed in my presentations, chances are greatly enhanced with a bigger haystack.
Thomas, would you like to comment?
I just wanted to add one little comment to what Christian just said, because I think it's interesting to see that with the huge chemical spaces that we're working in right now, the retrospective validations, which is often the only thing that you can do, becomes harder, because it is no longer certain that your known actives really are the only actives in that very, very big haystack. And then sometimes you get decoys that are wrongly ranked, but if you look at them, it's really hard to believe that they would not also be actives. But of course, nobody has the ability to do all these tests here, and I think that is a complicating factor when you're working with billions of molecules.
I guess we're increasingly reliant on the kind of positive feedback that Carl alluded to during his presentation. But maybe a related factor is how do we reassure ourselves that we're navigating into chemical space where we have freedom to operate? Is that something we can even control for during the kind of size searches that we're talking about here? Maybe I'll ask Carl to comment on that point first.
I'm sorry, Dave, can you repeat that? You were breaking up a little bit.
Sure, no worries, Carl. My question was whether there's any mechanism by which we can incorporate any understanding of navigation into new chemical space, specifically novel chemical space, during the course of the kind of searches that you run, Carl, and similarly to Christian.
So you're talking about like the freedom to operate is what you had basically said.
Exactly.
Yeah. So the way that we kind of do this is you can use obviously SciFinder and open patent databases, but the way I've done it in the past to see the novelty and if anything has actually been prior art is I've downloaded all of the patent literature in XML, and then I parse it, change it into actual molecules, and then do overlap and substructure searches around it to see if there's anything that you know, lights up. And if it does, then you take it further to going into like a SciFinder search to see, and then identifying the patent. Unfortunately, we can't do that free. I know that the team has been working on trying to figure that out a little bit more. But the key point in this is if you were to create your own space with our building blocks, it would give you more of an advantage considering that you probably have already looked at the scaffolds and templates that you're already working on.
Christian, would you like to add anything to that?
Yeah, the freedom to operate or the IP value of your compounds at hand, that's another interesting and tricky question, I would say. In fact, a couple of pharma companies have contacted me after we introduced this concept of the virtual chemical spaces, because all of a sudden there was this concern, so your machinery can now theoretically enumerate all these billions of molecules. In fact, as we've seen from Carl's presentation, Galaxy has also been enumerated. So you get into this range of billions of molecules being enumerated. And for quite some time, the rule of thumb was, okay, once it's drawn, once it's out there, it's kind of published already. It's tainted. It's not so sure anymore if I can claim ownership of the molecule, because the concept in the past was, well, as soon as you draw it, as soon as you design it, it's kind of yours, unless somebody else first publishes it. And those lawyers I've been talking to, I told them, well, you got to rethink your general strategy, because a molecule that can theoretically be enumerated, and you have seen here in my presentation, 10 to the power of 26 molecules. In theory, the algorithm can find any one molecule in there. Have they been published? I wouldn't say so. So I think IP value starts only later in the process. You cream off the top with your virtual screening strategy, and then you do an assessment. Is this molecule, has it been published before? And are the properties also exactly what I need? So that is kind of pushed forward into the later phases. And just one other comment. One thing that I observed is that what's sufficient is one proprietary building block in your molecule. and 1,000 reagents on one side and another 1,000 reagents on the other side of this proprietary building block may constitute 1,000,000 molecules, but they are all proprietary just because this one piece is your proprietary piece. Keep that in mind as you think of these millions of molecules. Yeah, just one bit of it being proprietary is already sufficient. So I think in these huge spaces, you also have a lot of opportunity to enforce or to reach the freedom to operate.
Christian, I wonder, in your topological maps that you build as the starting points for your models, very often in medicinal chemistry, we have an understanding of where in a molecule we have some wiggle room, if you like, and which pieces need to be quite fixed. So what kind of sensitivity there is around particular aspects of the molecule. Are you able to tune different elements of the map for more or less diversity of features as you go into the search?
Yes and no. The descriptor is as fuzzy as you've seen it. It's just a blob on the topology map saying has an acceptor, is a ring structure or not, has a certain size. What you can control in a feature tree search is you can mandate that a particular part of the molecule has to be mapped with higher precision. That doesn't mean that the substructure needs to be identical in the hit molecule, but it means that the similarity on the pharmacophore property level must be higher. So what this means is that if a molecule is in your space that has exactly this particular scaffold, let's say, that you also have in your query and you want to lock in on it, well, you just mandate that the similarity must be as good as possible. And then you greatly increase the chances to find molecules that have that particular scaffold and that are more diverse or less similar in the periphery, for example.
A question to both Carl and Christian. I wonder if during the execution of a search on this scale, you have the opportunity to bring in other predictive markers that might direct you towards compounds of a particular predicted distribution or stability or ADME property profile. Are you able to bring in other kinds of predictors during these activities?
Carl, if I may go first.
Go ahead.
So there are two categories of predictive models. One that relies on the whole molecule to be presented to the model, and only then the model can do its prediction effectively. And other models that work also on partial structures, like the presence or absence of a particular substructure, maybe a potential toxicology issue or so, or simple additive properties like the number of stereo centers, the number of acceptors, donors, these kinds of simplistic and additive properties. The ones that work only on the whole molecule, they can only be applied afterwards, because only afterwards the molecule is complete. The models that work also on partial structures, they principally also work during the search. So if there is a substructure with a tox issue, I can find that out earlier than afterwards, because it can be present also already in one of my building blocks that together compose the whole molecule, or a parameter like the size of the molecule is simple additive. I can control it during the search to stay below a certain limit. So these latter, more simple, more additive parameters, they can be controlled during the search, but the whole molecule, models, descriptors, calculators, well, they can be only applied as a post-processing.
Understood. Thomas, would you like to contribute to this aspect of the discussion?
Just, I felt it almost sounded a little bit pessimistic what Christian just said, because my feeling would be that the majority of things that a med chemist looks for when they are searching for a drug-like molecule are these relatively simple additive, number of rings, number of stereocenters as a cue for synthesizability. So I think we are doing quite well in filtering the libraries to whatever property distribution we want for most properties that are of interest. So.
Perfect. Carl, did you have a comment here as well?
Well, the way that I usually approach this is just a quick comment is it's real easy to build the spaces. So you can also filter pre on the building blocks for any kind of structural alerts, etc. But as Christian said, the majority of it is done post searching. And the way that I use it, I mean, I utilize NIME a lot to do all of the predictions post and pre when I build spaces. And your spaces can be built to your liking. So you don't necessarily have to use the whole galaxy space. We also provide the building blocks and the scaffolds. So your pre-filtering can happen then as well.
Got it. Okay. Of course, in other areas of finding, we're also working with massive real libraries. And the obvious example is with DNA encoded libraries. Have you done any comparing and contrasting of the successes of searching large virtual space using a platform like Infinity versus a real screen of a library like a DNA encoded library? Do we have any feeling for the diversity encompassed in a Dell library versus the massive libraries that you're screening virtually? Christian, would you like to have a go?
Yeah, sure. In fact, we have encoded Dells already also as virtual chemical spaces. The need to explore a vast Dell library is a bit of a complicated thing in itself, because a Dell also can contain 10 to the power of 12 molecules, and simply figuring out A, what's exactly in there, and B, what's in the vicinity of a particular molecule is an interesting question in itself. And I would say one can be considered the wet lab experimental approach that complements the virtual chemistry experiment in the computer. I would argue that with the size and the number of spaces that we have coded so far, you have seen these this large number of spaces that we have coded together with different pharma companies. We cover quite a diversity of different reaction types and space topologies, you name it. I'm not sure if Dells cover such a broad spectrum, but they certainly give also access to billions and beyond molecules. And that's the big advantage on an experimental level. Yeah, so what's in there and what you find as a hit has really some chances to be a real hit. So I like to see not it to be a competition one against the other, but actually a complementary approach, a little bit like HTS and high throughput, and virtual screening also can complement each other.
A question for Carl around the diversity of the galaxy library. Have you done any calculations that try and understand the degree to which the chemistry space that's already validated and the building blocks available to the galaxy team, does that limit diversity? Are there obvious directions of further diversity that you can build into that library in future generations?
Yes, you can definitely build in more diversity as the chemical space of commercial compounds become more available. You can create your own commercial slash custom spaces, which will actually build out the diversity. However, Wuji's team, their building block and scaffold team are constantly searching for new cores, new building blocks in the literature, JMed Chem, all the way out to the patent literature. So I think the diversity is only going to increase as we even get more and more diverse chemical spaces as new and novel building blocks and scaffolds become available. And just by doing an analysis of the building blocks that Wuji has, the overlap in the entire chemical space is very minimal. So just on a building block to building block comparison for the other virtual libraries that are out there, such as ChemSpace, Enamine, the overlap is like less than 2%, just based on building block and scaffold comparison.
Okay, thanks, Carl. Christian, and maybe Thomas, this is also a question for you. When you're looking at these kind of techniques, do you find they work particularly well with different kinds of target classes and not so well with other kinds of target classes? Would you like to start, Thomas?
I was about to say I'm not so sure that can be really very clearly said. I would more say that depending on the target class that you work on, the questions that you're asking are probably quite different. I mean, I think, let's say, if you're in a kinase space, you'll think a lot more about selectivity and prior art and existing kinase inhibitors compared to if you have a truly novel and not well explored target. But I'm not sure how well that translates to a question of, okay, how to put together the library or how to search through the library. I think we're a step ahead of fully understanding the peculiarities of your specific target at the stage that we're talking about.
Okay. Christian, any feedback from you?
Yeah, I agree with Thomas there. I think the target very much dictates which kind of virtual screening technique applies best for that particular target. And as I've showed in my presentation, we are still in the process of building all these capabilities. Yeah, it's similarity searching for a start. Well, that's great. It gives us an option to explore within the billions now. Substructure and fingerprint searching next, so more closely related molecules can be found then, or something around a particular scaffold. And then docking as a next stage. And once this arsenal of possibilities to explore the billions of compounds is more complete, then it is just a matter of, okay, which method is best applicable. And then I would argue it works for any kind of target. Yeah, it just increases your chances since it makes the number of options so much bigger. Yeah.
Thanks, Christian. As we work increasingly with machine learning based tools, and we start to think a little bit about applying generative models in drug discovery to perhaps drive us into new hit finding or scaffold modifications, do you think the kind of platforms that we're discussing here today can in some way be melded with those kind of generative tools to create something that benefits from both paradigms?
Yeah. Do you want to go first, Thomas?
I would just say, I mean, I would argue that, that's almost a loaded question, because these generative models, they breathe having a lot of chemical input data. So having something like, a DL library or an enumerated library or something that is a huge chemical space of stuff that makes sense. That's a perfect environment to train the generative models in, so that they are able to pick out, let's say, the holes that still exist in your chemical space and to fill them up. So I would say that is a good match, and these are technologies that will not replace each other, but are quite synergistic in most cases.
I also see it such that different machine learning techniques can help and complement what we provide at different stages of a development challenge or project challenge. So at the very beginning, you need, in my opinion, mostly rapid turnaround times, and there machine learning can come into play to harvest the information that you gained from the last round and make the next pick of compounds most effectively, so that the machine finds better and better molecules in these big spaces. There, I would say machine learning is rather for post-processing of the results and harvesting from the experimental data that you gain, and the aim is to do it quicker and quicker and quicker. Yeah. In later stages of a project, where you you can think of it like with the spaces, we lay out a general map of possibilities. In the end, these all will only be starting points for the real discovery projects, maybe getting you so and so far, but at some point you will have to get off that map and be particular, do specific chemistry. And their generative models may support in more specific modifications, do a methylation here, and so on and so far, where it is really into the fine tuning and supporting the optimization cycles. So early on and later on, there's certainly aspects that can well be supported with AI methodology. That's my opinion on this.
Perfect. Thanks a lot, Christian. I'd like to give the three panelists here a chance to ask each other questions, if you have any for each other, or to make some closing points. Perhaps I'll start with you, Carl. Do you have any questions for Thomas or Christian?
Well, I kind of work with Christian a lot. So, I mean, any questions I probably have already asked him, and no, nothing for Thomas either. I just want to thank you for putting this together, and for Christian's presentation today, and I'm looking forward to Thomas's tomorrow. And thanks everybody for joining.
Thanks, Carl. Christian?
Yeah, likewise, I know the galaxy space. we helped build it. So no new questions at that end. And Thomas, I look forward to your presentation tomorrow. I'm sure questions will come up then again. And my last comment is maybe a little bit related to the last discussion topic, namely those AI models and getting finer and finer into the real chemical space. We have been asked numerous times, well, these big spaces, don't they overlap a great deal? In other words, is a lot of this redundant? And the fact is, it is not. And again, I'm coming back to this observation that a single modification or a single different fragment in a molecule that can have, say, 4 components or so is sufficient already to make it unique. We did a study a way back to compare one of these proprietary pharma spaces with our public domain space with a compound vendor space. And despite the fact that all these spaces are 10 to the power of 10 or bigger, so amazingly big, the overlap was minimal. And I found this astounding. I would have thought, there's at least thousands of molecules, if not millions, among the billions that are present in either space. But then I thought, typically people say that chemical space really covers 10 to the power of 65 or even more molecules. And if you consider that, then even the vast space from GSK, 10 to the power of 26, is just a drop in the ocean. And with that picture, it is clear that we are still only scratching the surface.
Yeah, I think it's a timely reminder that a lot of our screening collections in different organizations are as much a reflection of the historical biases of the chemists in those organizations as they are of the ambitions to build much larger screening decks. Thomas, would you have any closing comments?
Yeah, no, just to, as I said, I think it's very nice to have the virtually enumerated spaces because they are not subject to these biases of, okay, what do chemists like to make most? And I think we have consistently shown going to hundreds of millions, to billions, to tens of billions of molecules, you do find a lot of novelty and a lot of diversity. So this is not just a fallback to, the 1980s, before my time, combinatorial chemistry, which is A times B times C. There really is a lot of additional chemistry out there that this technology helps to explore. Yeah.
Thanks very much, Thomas, and I look forward to your talk tomorrow. So we're out of time. Thanks everyone for your discussion and Q&A. Thanks also to Christian and Carl for your presentations. I hope many of you can join us tomorrow for our next session when Thomas will come back and talk about Schrodinger's molecular design platform. And my colleague Li Xing will talk about virtual screening and machine learning applications to large data sets. Thank you very much, everybody. Good night.